Hi everyone, I'm one of the 37 trillion cells that make up you. Today I want to talk about meiosis. Most of the cells in your body are somatic or body cells and have diploid or the normal number of chromosomes. However, the cells that we care about today are your germ cells, which have half the number of chromosomes, and so they're called haploid cells. You may ask, why do we need cells with half the number of chromosomes? Well, the answer is that our germ cells, also known as our sex cells, are needed for us to reproduce sexually so we can create genetic diversity. Everyone has a different combination of DNA from his or her parents, so they are unique. Just look around you and you can see genetic diversity everywhere. In populations, genetic diversity is the reason species are able to evolve and survive. Germ cells have half the number of chromosomes as a regular cell so that each parent can contribute DNA and the offspring will end up with a normal amount of DNA. If this didn't happen, the amount of DNA people would have would double each generation. Every germ cell is unique and has a different combination of the parent's DNA, while each somatic cell is genetically identical. Now let's learn how meiosis creates 4 haploid cells from a single diploid cell. The process of meiosis is split into 2 different processes called meiosis 1 and meiosis 2. Meiosis 1 is the splitting up of homologous pairs, and meiosis 2 is the separation of sister chromatids. At the end, there are 4 cells with half the number of single chromatid chromosomes as the original cell. Let's slow down and take a better look at this. So let's say an organism has a total of 2 chromatid chromosomes in a normal somatic cell and it's going through meiosis. During meiosis 1, the chromosomes line up in homologous pairs. Homologous pairs are 2 chromosomes that have the same genetic information. Then the cell splits in half, creating 2 cells. Each new cell has 1 chromosome from each homologous pair, so a total of 4 chromosomes. Then, during meiosis 2, each chromatid chromosome splits. Each of the 2 cells created during meiosis 1 split again, so there are now a total of 4 cells with half the original amount of the genetic information. So now that you've learned a little bit about meiosis, let's learn exactly how it creates genetic diversity. The 3 processes that meiosis creates genetic diversity through are crossing over, independent assortment, and segregation. Crossing over is when chromatids from homologous pairs cross and trade genetic information. So looking at this pair, which has the same type of DNA but different variations, we can notice that they have traded some genetic information, so each chromatid is a combination of genetic information from both chromosomes of the homologous pair. Independent assortment is also involved with pairs of homologous chromosomes. It is when the pairs are randomly split into the 2 different cells in meiosis 1. In a homologous pair, 1 chromosome came from the individual's mom, the other came from the individual's dad. As a result, each germ cell is a random combination of chromosomes from each parent. Finally, segregation is just the separation of the pairs of homologous chromosomes. It means that each pair gets split up so the cells end up with half the pair. Whoa, that was a lot to learn about, so let's review. You learned that meiosis creates germ cells and helps create genetic diversity through 3 different processes. Ultimately, meiosis is what helps make you an individual. Thanks for learning about meiosis with me today.